Genetic information is held inside of the nucleus. It is found in chromosomes. Chromosomes are long strands of DNA that are tightly wrapped and coiled. DNA is a double helix shape that consists of phosphate, deoxyribose sugar, and a base group. The DNA is wrapped around proteins called histones. Within DNA, there are 4 base pairs that are each complementary to one another. RNA is similar to DNA, only that it is like one backbone of DNA. The base pairs for RNA are the same as DNA, only that instead of thymine, you will find uracil. Instead of a deoxyribose sugar, you will find a ribose sugar. So the hydroxyl group on the second carbon is on the bottom of the plane instead of the top. These strands are created in the transcription stage and used in the translation stage to be read to create proteins. Transcription is a process that involves RNA polymerase and the mRNA strand. The DNA strand is unwound and unzipped by the RNA polymerase. This exposes the bases, so they are now free for other nucleotides to attach to them. The RNA nucleotides attach to the DNA template strand, forming an mRNA strand. Then mRNA leaves the nucleus through the nuclear pores and travels into the cytoplasm. There are several signals within introns that are essential in the splicing process: GU, A, and AG. The GU and AG site lets the spliceosome know where the beginning and end of introns are. The first step involves 2 spliceosome complexes that bind to the GU site. This causes the RNA to loop, and now the GU and A sites are in line, and so they bind. The spliceosome undergoes a conformational change that separates the AG site from the exons. Lastly, the exons on either side of the introns are joined together, and the separated introns are then disintegrated in the cytoplasm.
The molecule involved in translation is called transfer RNA, or tRNA. The structure of tRNA consists of a point of attachment for the amino acid, sections of paired and unpaired bases, and an anticodon loop, which contains the 3 bases that make up an anticodon. Since genetic coding is degenerate, each tRNA molecule is specific to one amino acid and has an anticodon also specific to that same amino acid. tRNA is made from a single polynucleotide chain, and it's the smallest molecule compared to DNA and mRNA. It's clover-shaped, made of ribose, and contains uracil instead of thymine. Like mRNA, it's made in the nucleus but found throughout the cell. Now moving on to the stages of translation, or polypeptide synthesis. Translation happens at a ribosome, found on the rough endoplasmic reticulum or freely in the cytoplasm. The ribosome will attach onto the newly formed mRNA molecule at the start codon, AUG. The tRNA molecule, with the complementary anticodon sequence, moves onto the ribosome and pairs up with the codon on the mRNA. The process repeats as the ribosome moves up the molecule, bringing more tRNA molecules. 2 amino acids on the tRNA are joined by a peptide bond using an enzyme called peptidase and energy provided from hydrolysis of ATP. Eventually, an amino acid chain is formed, and when the ribosome reaches the stop codon, the chain can fold into a protein in the following ways. Right now, the protein is in its primary structure. It can then coil and fold to form its secondary structure. The secondary structure will then fold again to form the tertiary structure, and the linking of different polypeptide chains and non-protein groups will form the quaternary structure.